Good afternoon. I would like to try to explain our lab using explain everything, and we'll see how this works, if it's successful or not. But if you could please turn in your lab to page 82. This was where we left off last week in lab, and we provided you with the proposed experiment, and then you were supposed to predict the outcome. So just to remind you, we are basically taking two bacteria, either JM109, which is one strain of E. coli, or E. coli HY842, and we are mixing these two bacteria and allowing conjugation to potentially take place. And so what we're looking for is whether or not conjugation took place between these two bacteria, and the way we're going to do that is by plating them on plates that have a particular antibiotic. So what we're really trying to assess is the ability of JM109. Is this going to be able to transfer the plasmid, as shown here, the ampicillin resistance plasmid, to HY842? Let me see if I can do this. All right, so if JM109 is able to do that, it's going to have the ampicillin resistance gene, and on this ampicillin resistance gene is also the information to how to make the sex pilus. So the sex pilus is going to get extended, and then this plasmid will get replicated. It'll get copied, and then a copy of that will get sent to HY842. Such that when we're done, what we're trying to select for are bacteria that look like HY842, but now what they've done is they've been able to, let's see if I can move this. And so now the HY842 has taken up this plasmid, and we can maybe mark this as such that it is slightly different than the initial HY842 plasmid, because now this particular strain is not only resistant to zeaxanthin, as initially it was on its chromosome, but also it's taken up the ampicillin resistance gene. So now on the homework, what we asked you to do is to determine what was going to happen to these three different populations, each either the JM109 or the HY842 or the HY842 that have undergone conjugation. And let's see what happens when we place them in the presence of particular antibiotics. So if we have bacteria, the JM109, and there's no antibiotic selection, and we take a certain amount of that antibiotic or that bacteria and put it on a plate with no selection, then what we should see here is a lawn of bacteria. There's nothing to kill any bacteria, so they're going to all thrive and do well. Now, what's going to happen if we place the JM109 in the presence of the antibiotic zeaxanthin? So if you recall, the JM109 was resistant to ampicillin, so it can survive fine in the presence of ampicillin, but it cannot survive in the presence of zeaxanthin. So now if we treat it with zeaxanthin, you should see no colonies. However, in the presence of ampicillin, it'll be fine. It's got that antibiotic resistance gene, so we should see a lawn. Now, what about when we add zeaxanthin and ampicillin? Well, that zeaxanthin, again, is going to kill off the bacteria, so we should see no colonies. All right. Now, what about the HY842? So now we are looking at this particular strain. We know that it's resistant to zeaxanthin, and so the zeaxanthin is going to have no effect, so we should see a lawn in the presence of zeaxanthin. With no antibiotics, likewise, we should see a lawn. However, here with ampicillin, that should kill off that parental strain, so we should see no colonies. And the same is true when you're using a combination of the antibiotics. Likewise, we should see no colonies. All right, so now the question is, what do we get when we do a mixture of these two bacteria? So I think the important thing to keep in mind here is, with regards to this third experiment, what you've done is you've taken a a subpopulation of the JM109 and a population of the HYE42, mixed them together, and then poured that on a plate. So when you've poured that on the plate, what you actually have are, you have some JM109, you have some HYE42, and you've got those that actually underwent conjugation, the HYE42 conjugates. So we've got a population of all three bacteria on that final plate. So when we have no antibiotics, we should absolutely see a LAN. Nothing is dying. Now, what's going to happen when you treat with zeaxanthin? Well, when you treat with zeaxanthin, if we have the conjugates, they're resistant to zeaxanthin, so they should survive. The initial parental HYE42 parents should survive. However, the JM109s should all die. But the way that's going to look like on a plate is that's still going to look like a LAN. Okay, what about when we treat with ampicillin? When we treat with ampicillin, those transconjugates again are, and they're resistant to ampicillin, so they should survive. The JM109 parental strain should survive. However, the HYE42 should die off. So what we'll see here again is likewise a LAN. Now the key for the whole experiment is what happens when we have both antibiotics on the plate. So if you've got zeaxanthin on the plate, you are going to kill off the JM109. If you have ampicillin, you're going to kill off the HYE42. However, if there are trans conjugates, they're the only ones that will survive on this final plate. And so here, what we should see are countable colonies. So the way that we know that conjugation took place is really by, you have to have all 12 of these controls, but the key experiment is this final one here. When you mix those bacteria together, if we can see countable colonies, then we know conjugation took place, because when you compare that to either of the parental strains, there were no colonies under those conditions. All right, I hope that's helpful.